Oh,
Okay.
great. Awesome. Hello, everyone. So for those of you who may not know me, I am Paula Montero Llopis. I am the director of the MiCrOn core at the Harvard Medical School. As you all know, the mission of the core is to empower the research at HMS, and what we try to do is bring the technologies or techniques that will improve your research. And when we host seminars and we host demos for equipment, is because we have identified a need that is not being met, and we want to be able to get your feedback. So I am really happy that all of you came. Parhelia will talk about the system, but what I wanted to mention is we are going to have it in the core for a month, not a year, a month, and so we invite you to come and test it, right? So when we make these decisions, we want to make them together with you. And with this, I want to introduce Michael Kraft, or you want to first start with?
Yeah.
Okay, cool. So then Bill, so you are the sales rep here in the from Parhelia, because I've been just talking with you and see. Yeah.
I can introduce myself.
Yeah. So Bill.
Great, thank you.
Oh, and before anything, I would like to thank Parhelia for being so kind to provide lunch today, and they also brought some swag that we all like it. So please feel free.
Okay. Good morning and welcome. My name is Bill Kennedy. I'm your local rep here in Boston for Parhelia, and we're really happy that you're joining us here, whether online or in person. And thanks to Paula and the MiCrOn core. So, like I said, my name is Bill Kennedy, and I actually started my career here at Harvard Medical School. I graduated from college. I needed a job, and so I answered an ad at the time. It was in the newspaper, in the Boston Globe. I had no idea what I wanted to do, and I was basically answering every ad that I could. And one of them was for the Division of Comparative Pathology at the Primate Center, which is now defunct. But that was my introduction to histology, immunohistochemistry, tissue pathology in general. And it's turned into this wonderful career that I could never have imagined was possible. So I'm super grateful to be here talking to you right now. So, we're a microscopy core. This is in a microscopy core. So I wanted to put some images up here, some really nice images. We have one from a geomics protocol, a phenocycler protocol. This one's multiplex immunofluorescence with Opal. And then there's an RNA scope protocol. And so when I look at these images, for me, as somebody who is trained on the bench, who's done all of these manual stains, this represents to me lots of effort, time, and money that goes into developing these protocols and running them on a daily basis. And I know all of you here know what that's like. So, when we think of spatial biology and spatial biology protocols, we think of, we get a tissue block, or maybe cells. It could be FFPE, fresh, frozen, whatever. That time from the time when you get that sample to when you do that analysis, there's all these steps in between. And they can take days, or it could take hours or days. And in the clinic, a lot of these assays have been validated on automation. So it's just press a button, it's done from start to finish. In spatial biology, this is such a new and burgeoning field that we're not there quite yet. So what I hear from a lot of researchers that are doing spatial is that there's really two issues. There's issues of throughput and there's issues of variability. And so, you know, I talk to people all the time who tell me that these protocols take so much time. They take my techs all day, may take a couple of days. They're married to their timer and they can't do anything else. So that's one thing I hear a lot from customers. Another thing is people are concerned because there's variability in their assays. So there's variability from run to run. There's variability within the same run, variability between users, and this variability, it's a concern in and of itself. But then it also is compounded when there's so much variability that they need to repeat their experiments. By repeating their experiments, they're costing a lot of money. So could be anywhere from, you know, $100 on the lower end to $5,000–6,000 for a slide on the higher end, perhaps more. So this is a real issue, this repeatability. And coming with that as well is there's, with that variability, there's also comes what happens if the person who's an expert in that particular assay isn't there that day, if they're out sick, or, you know, even worse, if they leave, and who's going to fill their shoes? So that becomes a real problem for labs. And then the last thing I'll talk about here that I hear from a number of people, especially if they're running their lab as some kind of business, whether it's a CRO or a core lab, and they're getting complaints from customers who are telling them why is this taking so long? And so, you know, these are all issues that I hear on a regular basis from customers. Ideally, we want that, everybody wants that complete automation of these workflows. And so, you know, that's really kind of, at the moment, it's the holy grail of spatial biology in a lot of ways, from the staining aspect. The founders of Akoya knew that there was a need for this complete workflow. Our CEO, Nikolai, and Yuri Goltsev, they were the inventors of Codex, now known as PhenoCycler. So they're intimately aware and they understand the issues that are faced when doing these manual stains. They set out to create an instrument that was able to do all of this whole workflow, really everything from that H&E, I mean, from dewaxing and antigen retrieval, all the way through, you know, antibody incubation, hybridizations of probes, really anything. So they created this instrument called the Spatial Station. And so the Spatial Station is a brand new technology that's here in the MiCrOn core for people to come to check out. And it's really an instrument with a small footprint. It's benchtop, but it has a big impact. It will have a big impact on your workflow. It does that true end-to-end automation. So it does everything from that dewaxing, antigen retrieval, all the way through to the end of your protocol. It's easy to use, and that's an important point. It's easy to create protocols. There's pre-existing protocols that are on the instrument itself, but then you can edit and change these protocols any way that you want. It does heating and cooling, so it heats, so you can do your antigen retrieval. You can do your hybridizations at 37 or 45 degrees, but then it can also cool. So how many people here do antibody incubations at 4 degrees overnight? Anybody? And we have about, I don't know, half the people in the room here said they do. And so that's something that I've been asked throughout my career working with automation. Can we do 4 degrees overnight on your instrument? Up until now, the answer has been no. So you could do that, that incubation on the spatial station. We use small reagent volumes, so about 100, 110 microliters per slide per dispense. You can adjust those volumes. And one of the really cool things is that it does mixing for you. So you cocktail your antibodies, or if you want to do any kind of dilution series, you can do that on the instrument itself. And then last but not least, it's vendor agnostic. So you can use basically any technology that you want to on this machine. If you think it's going to work, try it out. We have protocols that we know work on the instrument. The vendors that we know, we have protocols that do work, but it's wide open. You can do whatever you want with it. Here are just a few examples of applications that work on the spatial station. As I said, the inventors of Codex, that was the first application they put on here. It also does DAB, IHC. You could do any kind of chromogenic IHC you want. You could do H&E, special stains. We have RNA scope. I know I've heard from a lot of people, SciSif is something that you do a lot of. It can do that. Multiplex immunofluorescence. Opal can do IMC. It can also do geomics staining, and just it can do a lot. This machine can do a lot, and so this is just kind of a high-level overview of the instrument itself. And so I'm going to hand it off to my colleague Michael Kraft. He's our field application scientist, and he's the one with some deep knowledge of the instrument. And so I'd encourage you after this, if you're interested in using it and implementing some protocols, please talk to Michael, as he's the expert. Thank you.
Perfect. Well, yeah, thank you, Bill. Thank you, everyone, for being here. Thank you for everyone that's online. And thank you, Paula, for hosting and helping us set this up today. So as Bill said, I'm Michael Kraft. I'm an FAS with Parhelia, and I'm really happy to talk today about the spatial station. So I'm going to give you an introduction about the instrument. We're going to talk about some key features. I'm going to show you how we set up protocols, and then I'm going to show you some staining. So while I go through this, I want you to really think about your own assays that you do, and if you're doing stuff manually, how you can use the spatial station to automate what you're doing. So you can set it on here, press start, and walk away, and then go do some other work or whatever else you want to do. So yeah, a little bit about the instrument itself. As Bill mentioned, maybe I'll just keep wiggling this. It's small but mighty. It's 2 by 2 by 2 feet. Fits on a bench top. It's about 85 pounds, so you can move it around if you need to. Once we install it, it's not set in a corner forever. There's a flexible range of pipetting from 1 to 1,000 microliters. So if you're doing small volumes like antibodies, like cocktailing, you can do lower volumes. If you want to do cell pelleting, like aspirating, things like that, you can do up to 1 mil, which is nice. And then everything in between as well. Yeah, as Bill mentioned, heating and cooling have a range from 0 to 99. So you can do those high temperatures if you do antigen retrieval. And then you can also do antibody incubation at 6 degrees Celsius. And yeah, one of the coolest things I think is that it is protocol agnostic. So any reagents you want, you can put on the machine and run them. You're not tied into any company's kits or anything. So if you have your own hematoxylin, so you make your own hematoxylin, you could use this and pair it with anything else. So I just wanted to give an overview of all the labware modules on the deck, because there's a lot of different flavors. And they all have the same SBS footprint. So that's a 96 well plate, that footprint. All those footprints, they all sit on the deck. So just starting in the back, we have the thermal module with thermal sheath. So the slides would sit in this thermal sheath and it's incubated in that sheath. And there's some sort of pattern. And then in front of that is like a buffer plate. So you add like larger volumes of PBS or water, anything you're doing washes with. Here in the front left, we have our slide standing module. So you can have this on the deck if you're doing things at room temperature, or you can put it in the thermal module if you're doing heating and cooling. To the right of that, we have our P54 module, which this holds 54 antibody tubes. So you can use this to draw antibody out of here and then cocktail it into a 96 well plate. Behind that, you have a tip rack. So we have a few different types of tips, which I'll get into. And then behind that, you have a liquid waste bin with a cover on it. So the waste is all collected underneath the deck with the lid on. So there's no like exposed wastes or gases in the air. To the right, this black bin is tip waste. So the tips, once they're discarded, they'll come over here and they'll fall into the waste. And the instrument comes with 2 pipette heads, and then you can add on 8 more for a total of 10 if you want, but it usually comes standard with 2. So this is a touch screen on the front, which makes it very user-friendly. It's very easy to operate. If you can operate, an iPhone or an iPad, you can program this to run your protocols. You can essentially start by creating protocols from scratch. And the idea there is you identify your source, which would be like a 96 well plate. You'd identify your target, which would be slides. And then you identify, like you set the volume. So you say, I want to take 120 microliters of antibody and put it onto the target, which is the slides. And there's a lot more complicated settings, but that's kind of like the basics of how you would create a protocol. So yeah, you can create it from scratch, or a lot of times we'll have protocols that we'll send to you and install with the machine. And once you have protocols, you can edit them with ease. It'll show the summary of the steps, and you can disable steps, duplicate them. You can put pauses in if you need to do incubation. You can adjust the temperature. You can adjust all sorts of pipetting settings. And it's all done on the touch screen. There's no need for like coding or anything. So you can do all your optimization right here. It's really user-friendly. So now I want to talk a little bit about the pipettes inside the robot, because these are really interesting. So here I'm showing all 10, but you guys have the one with just 2 right now, which is still really good. And they move in the X and Y direction on these rails that sit in the back and on the sides. And then there's a motor that allows them to go up and down in the Z direction as well. This instrument, we partner with Hamilton Robotics, and they have this new Cori 2 pipetting technology. That means these O-rings on the pipettes are good for 40,000 tips. And if you ever get to that point, Parhelia is trained in service and can help replace those. But the good thing is these tips are compatible with all 3 tip types, P50, P300, and P1000. And those are the main tips that we have here. So the way that it picks up a tip is this metal stop disc actually goes into the tip. It goes down in the Z direction. And then this eject sleeve, which can move up and down, will move up and allow that tip to be attached to the pipette. And then when it's time to drop the tip, it'll go over to the waste, and that eject sleeve will come down and drop the tip into the waste. And there are sensors and everything, so if it tries to pick up a tip and it's not there, then it'll realize that as well. So just a few things like around the instrument. You only need 1 cord. It's just the power cord, standard 120 volts. There's a USB, actually there's 2 USB slots, and we primarily use this and a flash drive for file transfer. So you can import and export protocols here. You can export run reports and save them on your computer as a PDF, which is really helpful. You could also potentially, plug in a keyboard if you want to type out things on protocols, so you don't have to type on the touch screen. And you also have the potential to use a barcode scanner as well. So you can do most of your file transfer here. There is, an Ethernet port if you want to plug that into an Ethernet jack in your lab, but I think usually the USB is sufficient enough for all that file transfer. Okay, so that's like the instrument and the hardware. But now I want to talk about like the labware modules that are actually sitting inside. Some of them are holding slides, some of them are holding reagents. So there's a few different types. So the first is the ST12, which holds slides. ST, so standard throughput 12, because it holds 12 slides. So you can put this directly on the deck at room temp, or you can put it on the thermal module. And if it's on the thermal module, it stays there for the entirety of the run. There's a lid that goes on top that not only helps create like a humid environment and prevents evaporation, but it also can help with protection from light getting in. And this is a custom labware that we had Hamilton actually make custom labware definitions for us. And that allows the pipette to know the exact X, Y, and Z directions that the pipette goes in, and it pipettes onto the slide. So there are 3 shelves, they all come out, and when you're setting up your run, you're putting your slides on, you can take these slides out. And then the slides are laid on the shelves like this, and they actually sit in the box at an angle, like a 45-degree angle or so. And then the liquid is dispensed on top of the slide, or right below the label. So you might be wondering, how does the liquid actually stay on the slide? The answer to that is this cover pad technology. So this is kind of like our core technology that works for not only this system, but our previous system. So it's this glass cover pad. There are these green spacers. So they're on the underside of this cover pad making contact with the slide on the edges. So if we look at a cross section, here are the spacers on either side. You have the tissue sample in between. This is the flow cell that holds 100 microliters of liquid. So when the slide and the cover pad are set up in the ST12, the liquid is added from the pipette right here at this dispense point, and it will actually go into that flow cell, and that liquid will be held in that flow cell until the next liquid comes in and pushes that liquid through the bottom. So once you add your solution here, you can add it as incubation for however long you want. You can do a 14-hour incubation overnight. The liquid's not going to be disturbed until you put the next liquid in. So we have a video if that'll work. It's showing a blue dye being dispensed into a flow cell with that cover pad on top. And you can see the strips on the edges where the spacers are. So the pipette is adding at a constant speed, which we set in the instrument. If it goes too fast, it could spill over, but we've dialed all that in. So you might see a little bit kind of sitting on that top lip as it slowly fills into that flow cell. Actually, that leads into kind of my next slide is why we introduced this chamfered cover pad. So this cover pad has a chamfered edge, which is essentially like a 45-degree bevel. And the point of this is, it's kind of this lip that allows a pipette tip to come down and go in, and it lifts the cover pad. So the reason that you want to lift it is if you do have bubbles, you can lift it a little bit off the slide to release those bubbles. That's the idea there. So yeah, these chamfered cover pads are becoming our standard cover pads, and you could actually allow for faster pipetting speeds due to this chamfered edge. We also have these image-ready cover pads, which are really cool because you use them for cyclic staining, where at the end of your staining protocol, you just add, we have our own Parhelia mounting media, you add that into the flow cell for this cover pad, you take it off, and then you image it. And then if you have another round of staining, you take it off the imager, you put it back on, and then the first step would be like flushing out the mounting media with PBS, and then you do your second round of staining.
Just have opinions about that.
Yeah.
Because obviously, like, so it's what thickness of this is glass, this is a cover slip, the thickness of a cover slip.
Yeah, I have to check. It's very similar to a cover slip. We have some upstairs I can show you.
Yeah, that would be great. It's just I mean, I understand with tissues, in most cases you're gonna have that on the slide, right? But like, depending on like the spacers, like, and the thickness of your cover glass, and when you're inducing these aberrations, that can be also X, Y, Z. And then I don't know like how good the rating is gonna look.
Yeah, I think there was someone from, I don't know, your group or someone from Harvard that got their hands on these. They said they looked good with the
Yeah.
so far,
Yeah.
but maybe not for every purpose.
Yes.
Yeah.
I mean, I would like to compare that like to regular cover sequences, not because, for example, like people who use these chamber slides with cells, we usually don't recommend using them because of those spacers and like the fact that like now your image quality like is very reduced. And like the thing is like especially if we are talking about multiplexing, like in many cases you're actually using primary conjugate antibodies, your signal is already gonna be lower. So like inducing these aberrations that like reduce the signal to noise ratio is going to be bad, right?
Right.
For the detected so,
Right.
this is like something that like perhaps like during the demo that we can check and test if possible.
Oh yeah, definitely. That's why I was excited to show this, because I know it's something that we want to try here. Yeah, definitely. And the only, like, not downside, but you have to be aware that this does require a slower pipetting speed when you're adding liquid, because it's so thin, it doesn't have as much of a lip for the liquid to grab onto. But that we've built all that into the protocols.
And maybe you're gonna mention that already. And I was very excited to hear that you can also do cover slips, so tissue sample on a cover slip,
Yeah.
which is my preferred method.
Yeah. Yeah, definitely. We can definitely see if we can get that going while we're here too. Yeah. Okay, so that's about, the slides and the cover slips. So just to touch on, like, you have your slides in the ST12, this blue box. You want to put a thermal sheath on to do heating and cooling. And then you notice when this lid goes on, it covers up these holes. So it's like, okay, how are we going to pipette with this lid on? In the instrument, there's these two paddles that sit in the instrument, and the pipette actually goes down and attaches to these paddles and picks up the lid and moves it on and off. So when everything's added and you're ready to incubate, you just go ahead, lift the lid, put it on, and you're ready to go, heating and cooling. Then for waste collection, there's two options of waste collection on deck. This is the one at room temperature. So the waste container just sits directly under the ST12. So all the waste is collected in this box. And again, it's covered so it's not exposed to the air. You also have this option to add like a hose if you want to like vacuum out the waste, but we don't generate too much waste here. But if you have some sort of protocol that's generating a ton of waste, that might be applicable to you. So here are some of the reagent plates that we use. So typical 96 well plate. So we'll use this for small volumes, antibody solutions, things like that, around 150 microliters. This 12 trough plate, we'll fill it with buffers, alcohols, waters. And then this 96 deep well is kind of intermediate between those two. And say you have, you're doing 12 slides and you're giving all slides the same antibody like cocktail. You'll just do a bulk antibody and put it in one of these wells and just have it aspirate from that well. You could also do like flow cytometry work out of this 96 deep well, where if you have cell pellets at the bottom, there's a V bottom, and you can aspirate the supernatant, and then you can come over and resuspend that cell and set like the mixing step to mix that, resuspend that cell, which is really nice. So here's just an example of how you would set up like an H&E. So you figure out like the small volume reagents that you need, which are right here, and then you put them all into this first column, right? And then your larger volume reagents, you put them all into your 12 trough. And then if you had one sample, they would just be in one column. And then if for every subsequent slide you had, you would just repeat it for those remaining columns. And then sometimes people ask, if I'm doing 3 different antibody cocktails, are they going to go to the right slide? And so there's a slide order. So whatever you put in column 1 is going to go to slide 1 over here. Same with column 2 is going to go to column 2. So nothing's really getting mixed up across, especially if you have like, you could do 12 different antibody cocktails and they would all correspond to the same slide. So for protocols, there are essentially 2 options. Like I said, you can import a protocol, you can create your own. If you want to import one, we have a bunch that we'll send to you, or we have them when we do the install. You put it on this USB drive, you plug it in. The protocol files aren't that big, so you're not going to have to worry about storage space. And then when you go into this file manager tab on the software, you can select protocols. You can do things like export them. Like say you're having trouble with something, you can export it, send it to us, and we'll take a look at it for you. You can duplicate protocols. Say you have one that's locked down, but you want to duplicate one and then change it around, modify it. You can delete protocols, things like that. So there's a lot of options in this file manager page. So looking at a protocol on screen, this is actually a protocol that we set up last week at Standard Bio up in Toronto. So kind of similar, simple IHC assay where we set the antibody solutions in this 96 well and the buffers in the 12 trough. And when you're writing the protocol on the screen, you're simply selecting your solution. So here we're saying, okay, we're taking 3% blocking solution, 3% BSA. And we're transferring that to the slides. And then after that, we introduce a wait step. So that's going to incubate for 45 minutes. Then we want to add our antibody solution. And then once we add the antibody solution, we can put the lid back on where the slides are. And then we set the temp to antibody incubation temp, which was 4 degrees. And then the step after that is incubate 14 hours overnight. So it's as simple as just like writing the protocols here. You can edit, you can move these steps around and things like that. So it's fairly easy to create and edit protocols. And then when you're ready to run, you'll see this screen, which shows all the labbers that you need for this protocol. So you'll set up your assay on the deck. You'll get everything ready that you need. And the cool thing about this instrument is there's a camera on the ceiling. So you'll say detect when you're ready to run, and the camera will detect that all the labware is in the correct position. And if something's wrong or something's not in the right position, it'll ask you to confirm. And it'll also detect if there are any tips missing, which is nice because then you can use all tips. Like if they're scattered all over the place, it'll still use those tips. And then also if you look in this details box next to like this buffer reservoir, it'll show you like the location of all the reagents, like where the reagents need to go. For instance, a first or second well is 100% ethanol, and it knows that you need at least 900 microliters based on the slides that you're running. So this is very helpful when setting up the protocol. And then after it's finished, you get this nice run report that you can export as a PDF. So you'll get like reagent location, you'll get a summary, you'll get if there are any errors, like someone came by and opened the door and there was an error. You'll get all that, and then you can keep this for your files. And lastly, I just want to talk about the pipetting settings. So there's a ton of pipetting settings. Oh, go ahead.
About it's in a sense of what if the liquid take it out, so that we stop pumping air or automatic air.
Yeah, so there's a few options. So with the system, you can do, it does liquid sensing, right? So which you can turn on and off. So if you have it on and it goes into a well and there's no liquid, it's going to pause and it's going to give you a few options. It's not just going to abort the run and start, like just stop. It's going to say retry, which like if you realize you forgot a liquid, you could go pipette it in, retry. You could say like something like start from bottom, or there's actually an option that says air. Like for some reason it's having trouble detecting liquid. You're saying just aspirate it anyways. So it shouldn't be an issue if there's air. But what's nice, you do have those options where you don't just have to like, you could skip it too. Like you could say skip well. So yeah, air not a problem, but also it's nice to have those options where you can like open the door while it's in the middle of a run. Yeah, it's a great question. Yeah, so this is leading up to exactly what I wanted to talk about. Like, so with these black tips made by Hamilton and these black CLLD plates, so that stands for capacitive liquid level detection, with the modules and the liquid sitting on these plates and the tip touching the liquid, that's how it actually determines if there's liquid in those wells. So like I said, you can turn it on or turn it off. And then a lot of the settings are here for pipetting. The biggest thing is like the pipetting speed. Like for instance, like the chamfer cover pads is a speed of like 50 to 100 microliters per second. But the image ready cover pads, because they're so thin, is closer to like 7 to 10 microliters per second. And if you have like a really viscous reagent, you might even want to set it lower than this. But the good thing with all these settings, it can be a little overwhelming for the first time user, but like if you are using a really viscous reagent or something special, you can dial this in to make it work. Okay, so finally, I can show some staining images. So one of our most popular and successful protocols so far is Akoya's PCF protocol. Here we're just comparing manual staining versus staining on the spatial station, and we're looking at, you know, FFPE human tonsil. So all these samples, you know, got the same cocktail, and it came from the same block. I think they're just serial sections. So we want to compare them. You know, we want to look at the signal. Was their signal lost? Is there change in background? Things like that. So we noticed, you know, the staining is fairly consistent. There's probably stronger signal with the spatial station. Maybe the concentration could be dialed back, which again would save reagents and money. But generally, like if the staining is specific and there's no significant change in background, I think it looks good. And even if the staining is exactly the same and comparable, then I think it's still an advantage using the spatial station because it's going to pipette the same every time, and it's going to pipette for you so you can walk away while it's running. And again, Paula, this is your question about, you know, is there differences? Because here we have a tissue closer to the label and tissue closer to the bottom, and there's been tests to see if there's consistent staining for both. Because one might think, yeah, everything's going to be absorbed at the top. So again, we haven't seen it to be too much of a problem. Yeah, maybe if they're really large tissues or really thick tissues, it might be an issue where you're going to have to add double or triple antibody, but so far we haven't seen it be an issue. So that was Akoya's PCF protocol. There are a few others that we've been working on as well. UltiView's in situ-plex and then cell signaling signal star. So these are similar in that they use bar-coded antibodies as a multiplex IHC protocol. The signal star in particular, I have some experience with, and doing it manually is very labor intensive because it requires like 8 rounds of washing. And for those washing steps, there's like an 8-minute incubation for each. So you really have to be at the bench or be around the bench with your slides. So when we automated it, it was really good because we could just run it and walk away. Some challenges with this protocol is it got really long. It's close to 100 steps. And like I said before, there's a highly viscous solution for the amplification steps that we really had to dial in. So for the signal star assay, it's a 8-plex. There's 2 rounds of 4-plex. So here we're looking at FFPE on tumor samples using the chamfer cover pads and imaging on the Zeiss Axio scanner. So this is an assay that we're really proud of, and we're happy that it's been successful on the spatial station so far. And then lastly, we did some tests with the image-ready cover pads using the UltiView in situ-plex assay. So we wanted to compare versus like regular cover pads after 2 days, and after 3 days. Like, is there any drying out? Is there any changes in signal? And we found that the staining and the signal remained consistent even after 3 days using the Parhelia mounting media with image-ready cover pads. Which is a good sign because, yeah, imaging sometimes takes a while, and we definitely can't have things drying out underneath the image-ready cover pads. I want to thank Nadia and Noemi. They might be on the call, but they did a lot of that staining that you just saw down at the NIH at the NCI. Also really want to thank Kevin over at UltiView, who's done a lot of tests with those image-ready cover pads and the drying out tests, and just been a really good collaborator with us for a while now. So yeah, that's all I have. Thank you guys. We'll take any questions, and yeah, we'll be here for discussion as well.
Do you guys have any questions that we haven't covered? Or like some assets that you have in mind that perhaps you wanted to try?
Yeah.
Yeah, I think the Visium H&E is pretty locked down. We have that part of it. The rest of the Visium we're still developing. Yeah, I actually had some slides about the Visium H&E, but maybe I could get them to you after. Yeah. Yeah, we can share. And like that's the thing, all our protocols we're just sharing with people. Yeah.
This is a question that I have to this. We like to work with our trainees to any aspects of microscopy. The vendors that we have the most access is people who have the same mission statement as we do. So will Parhelia also be willing to help us troubleshoot issues or like set up like protocols that like people are doing manually to be able like to be used with this, or are we going to be doing it on our own, or like how are you pushing it?
Yeah, definitely. I mean, like that's what I hope to do. Yeah, like this is part of training, understanding how the instrument works. But until we actually like go from the manual paper protocol to the instrument, that's really when you start to learn how things work. So for me, like I consider that part of the training, and I want you guys to have successful staining. So yeah, definitely.
So 2 questions. One about the heating and cooling. Is it a Peltier heating and cooling system?
No.
So how fast does it get to the temperature?
Good question. It heats pretty quick. I think if you're trying to get down to like 4 to 2 Celsius, that takes a little bit longer. Yeah, it also depends like where you're starting from.
The slide doesn't move down until it reaches temperature.
Yeah, it's sitting.
Cover that's heating.
Yeah.
Yeah, it's heating from the bottom, and then the thermal unit is on top. Yeah, and we've also calculated because that plate is going to be different temperature from the actual sample temperature. So we've done a lot of calibrations using temperature probes, like what's the actual sample temp when the plate is this temp. So we factored all that in as well. Yeah.
Second question is, so it seems to me that once you've got a protocol going, especially if it's like a longer overnight kind of protocol, that the entire station is sealed. Is that right? Or
There's a door. I mean, you can open the door. It's not locked.
it's more in the question like we're in a multi-core user facility and no one's going to be synchronized with the other users. So if someone comes in with a 2-day kind of protocol, does that mean the system is used up for that 2 days? It can't. Okay.
Yeah, you can't really do like 2 protocols at the same time. Yeah. Yeah.
You have to worry about evaporation. Or is it so?
No.
So, did you mean like in the solutions?
Yeah, out of the little.
90% ethanol, 100% ethanol.
Usually no, and like there's things that we do at higher temperatures, like the enantiomer retrieval. Like if it's like a 40-minute at 97 Celsius, it's like very high temperature. So what we'll do is just like we top off the solution like every 5 minutes or so to just make sure it's not evaporating. And then for like all the reagents and stuff in the 96 well and the 12 drop buffers, we have different seals we put on top too. So the pipette tip can just go in and puncture the seals and get the liquid, and there won't be any evaporation. And then you could like put a fresh seal on, or if you want, and then put it in the fridge, so you can keep that and reuse it. So yeah.
The questions?
Yes.
Tell me about RNA stroke.
So we have it really well validated for our older system, which uses the same cover pad technology. Similar pipetting robot, but it runs on a Python script. So we're just in the process of developing it over to this protocol, to the system, which is simply just like writing it and getting it up and running validated. But we're confident that it's going to work, just like I said, because that core technology is the same. Yeah.
And when we're talking about RNA stroke, it's really talking about geometry out, sort of fluorescent.
I'm not sure exactly. Yeah, we can talk about that. I can get an answer for you. Yeah.
So if you don't have any questions, we can stop here. The instrument is in NRV 832 for those of you who use the course, like you know where that is.
If not, you can email us and then we can show you the instrument. We are going to get trained in how to use the system. As I mentioned, it's going to be here for a month. So if you want to think of protocols that you wanted to test, as you guys also know, we like to push the limits of instruments. So if you help us push them, we will appreciate that.